But, well, last but not least, we have Jessica Luer from NOAA GFCL. Jessica is interested in, calm down, everybody. Okay, please pay attention, thank you. Jessica is interested in plankton modeling, zooplankton size spectra, all the optical systems, lots of different cool things. So we're really lucky to have her here. Yeah, Jessica, take it away.
All right, well, thank you very much, and thank you to the organizers for inviting me. This has been a very stimulating, I've only been here for the second week, but it's been an extremely stimulating 3 days. So my talk is titled Mixed Feedbacks and Climate Impacts on Zooplankton Vertical Migration. And so you've heard throughout this active flux section, the components of active flux include the mesopelagic migrant pump driven by diel vertical migration, as well as the seasonal lipid pump driven by ontogenetic and copepod seasonal or zooplankton diapods, essentially. But here in my talk, I'm gonna primarily focus on the mesopelagic migrant pump, just because we know a little bit more about their drivers and how those drivers will change, and that they are a larger, the total flux is larger than the seasonal lipid pump. With apologies to Svenja, the timing of that pump also happens at a time in which the overall fluxes it's important to get the fluxes down deeper. So this is where I'm focusing my efforts in this talk. So what I'm going to do is split my talk into two halves, talk about the drivers of active DVM flux, starting with the first order driver, net primary production, and then moving on to the series of second order, what I'm calling second order drivers: optical penetration, plankton size structure, temperature, and oxygen. And the inspiration for the title of this talk came from Stephanie Henson et al.'s paper 2022, where they highlighted that this vertical migration depth and the vertical migrator biomass is a source of the uncertain feedback with atmospheric CO2. Is this a pointer? Okay, yeah, these loops here. Okay, I wasn't sure. I was afraid of touching other buttons based on what happened earlier too. And in the second part of the talk, you already heard a 3-minute short synopsis of this work, but I'll dig into this a little bit deeper. This is work done in collaboration with Laura Splandi and Mathieu Poupon, a graduate student at Princeton University, where we're modeling zooplankton DVM in an idealized double gyre simulation. All right, so going into the drivers. The first order, I would argue driver would be NPP. So Santiago showed us very nicely some work from the Malaspina cruises that zooplankton biomass throughout the water column into the mesopelagic and bathypelagic relates very strongly to NPP. And there's some really great work from satellite imaging from Barenfeld et al. 2019 using satellite-mounted LiDAR instruments looking at the differences between day and night backscatter to measure the extent of DVM. So what they're showing in this figure is greater extent of vertical migration and the oligotrophic gyres, as we expect. The zooplankton and other migrators are avoiding visual predators. And what they also found is that the flux of DVM is strongly tied to NPP. So this makes sense. If the zooplankton biomass is strongly related to NPP, then the flux would be as well. And this is consistent with what Santiago showed in his 2019 paper. And then, so this is from Westbury et al. This is a study that Mike alluded to yesterday. So what Westbury has done is combined the MPP algorithms, 3 different MPP algorithms, the VGPM, CBPM, and CAFE algorithms over a 16-year time period, and showed that on average that there's about 5 to 10% global decline and MPP already observed in the satellite record. And that decline is even greater, particularly in the subtropical gyres, so like the North and South Pacific subtropical gyres. So we've already seen this decline, but climate models are showing quite a divergence in responses to MPP in the climate projection. So this is from the CMIP-6 effort, and what the CMIP-6 set of models are showing is that some models are projecting an increase in MPP. And these vary quite a bit by region. So in Tagliabue et al. 2021, they broke down the contributions from different CMIP-6 models and showed, for example, in the North Atlantic subtropical gyre, so I think here, on average, the models are showing a decline in MPP, but there are some models that show very little change. So this may be due to some specificities and distinctions in each model. There was a nice paper by Laurent Bopp et al. Suggesting that, at least in Piscis, this may be due to diazotrophy increasing significantly compared to other models. So to first order, you could just relate active flux to MPP and say, okay, we're done. But that would not be as interesting because there's so many other drivers. So what I'm going to do is kind of dig into the second order. What I'm arguing is the second order drivers. So the first of those would be optical penetration. So zooplankton use light cues as what's called like a proximal cue to initiate migration, whether they're going up or going down. And this nice paper from Oman and Romanigan, 2016, showed that the DVM amplitude relates very strongly to optical penetration. And similarly, in this series, a compilation of studies of the cross-frontal studies from the California Current LTER, Powell and Oman show that in this example, the vertical, what they're doing is crossing a front, and there is much more chlorophyll productivity on this side of the front than the other side. So on the side with greater productivity, greater chlorophyll, and shallower light penetration, the zooplankton are migrating about 100 to 200 meters less in their amplitude. So this is a very strong effect. Now, some models have shown that optical penetration will change in the future. So this is a really nice study using the Darwin model, where they looked at changes in chlorophyll and then associated changes in the blue wavelength and the green wavelength. And what Steph Dukiewicz et al. Showed that in the North Atlantic and in the Southern Ocean, the oceans will become more bluer. So optical wavelengths will penetrate a little bit deeper and less green. However, in this recent study from B.B. Kale et al., looking at the different specific wavelengths from satellite observations, they're showing much more mixed results. So what I have here is, these are just the areas where the signal to noise ratio in the satellite observations are greater than 2. So here are the blue wavelengths and here are the green wavelengths, and what they're suggesting, and I think Steph Henson mentioned this yesterday, is that the oceans are going to be greener. So this is in contrary to these results, but regardless, using a combination of the satellites and the models, they're suggesting that the time of emergence can be quite soon. So we should be able to start seeing a signal that departs from the noise in the North Atlantic, in less than 20 years, which is pretty exciting. So, so optical penetration is a little bit of a mixed bag. We're not entirely sure what's going to happen, but we think that the North Atlantic will experience the largest changes. Next. Next is them working down to plankton size structure. So again, from Oman and Romanigan, 2016, they showed that both the depth and the amplitude of copepod diel vertical migration is strongly related to size. So the smaller copepods will be migrating to shallower depths and exhibit less migration compared to the medium and larger zooplankton, such that it's actually at the medium copepod size ranges that they exhibit the largest amplitude of DVM. So we've known for quite a bit that the effects, combined effects of temperature and stratification resulting in decreased nutrients is expected to also change community mean size. What we did recently in this study that's in preparation, led by Marco Corrales, my postdoc, is took a number of CMIP6 models. We chose the models that represented some amount of size in the plankton, asked the model developers to assign a size range to their plankton functional types, and computed a biomass size spectra. And what this is showing is the difference between projected changes under SSP5 compared to historical simulations. So the blue areas are showing areas where the size spectra becomes steeper or have more small plankton, and the red areas are where the size spectra becomes shallower or have larger plankton. So in general, with some variations, the models are saying that in the subtropical gyres and in the North Atlantic, the plankton community structure will become smaller. Whereas there's some models that show a pretty large increase in plankton community structure in, for example, the Southern Ocean. I think largely in this model driven by diatoms. So, but this is only just focusing on the crustacean zooplankton size structure. When we start to think about broader community structure, this can be more complicated. So in this recent study that we published last year, we introduced two new size classes of pelagic tunicates into the cobalt ocean biogeochemical model. So the cobalt ocean biogeochemical model has it has three size classes of zooplankton, and the two mesozooplankton are crustaceans. And so what we did is we introduced an appendicularian and then a south doliolid pyrosome. And one of the things that we found is that there was a relationship between the large tunicates that was different from the crustacean mesozooplankton, and such that at lower chlorophyll levels, their biomass did not decline as much as the crustacean mesozooplankton. So the implication for this is that under climate change, as chlorophyll decreases, the relative contribution of these large tunicates may increase. And these large tunicates are migrating quite deep. So this is a nice figure from Steinberg et al. 2023 showing the migration of salps in the North Pacific on their export cruise. And these salps, in our model at least, and also in reality, are competing very strongly with the microzooplankton and small mesozooplankton, which are migrating very little. So potentially with changes in the plankton size and community structure, you may get this complication that involves the gelatinous zooplankton. All right. Moving on to my last two, so temperature and then oxygen. So temperature, the relationship between metabolic rate for a whole community and temperature has been established for decades, from Epley 1972 and onwards. And what this may result in is that as temperature increases, then the metabolic needs may be greater than the food availability for many zooplankton, particularly in the subtropical gyres. So this will result in a pressure towards smaller bodied organisms, which I'll show you in my next slide, and increase excretion and egestion in the upper water column. The question of whether there will be deeper migrations due to this increased metabolic load due to increased temperatures is really an open one, and I don't think it's been settled at all. So this may be a complication, but we don't really know right now. And the very last of my drivers, I hope I haven't lost folks yet, but is oxygen. So oxygen serves as a hard limit for where heterotrophic organisms can live. And in this nice study, now 10 years old, from Daniele Bianchi, he showed that when you model vertical migration of zooplankton, they will increase and intensify oxygen depletion because they're migrating to just above the edge of the OMZs. And in this recent study by Curtis Deutsch, he compiled data from a range of organisms, not just zooplankton, but spanning the marine spectrum, and showed that the combination of decreased oxygen and increased temperature will result in pressure towards smaller body sizes in all marine organisms. But this story of oxygen is a little bit more nuanced, I would say, from the traditional understanding that climate change will result in decreased oxygen globally. In this recent study from Buseski et al. In 2022, what they did is they looked not only at the total OMZs, but separated by different thresholds. So if you look specifically, so actually just point your attention down to this bottom right plot. If you look at the OMZ as defined by, 120 micromole per kilogram volume, then yes, OMZs will increase. But then when you start to break it down under different thresholds, what they showed is that the core of the OMZ and at oxygen levels about 60 micromolar and below, then these OMZs may actually not expand. So this may actually result in some refuge for vertical migrators. And I'm not getting into fish at all in this, but the fish story complicates this a little bit more. So, all right, so those are the last of my drivers. And then moving on to the second part. You guys are all ready. So Matthias showed a preview of this, but this is work that we started a couple years ago that's funded by NSF, where we are modeling zooplankton vertical migration in an idealized double gyre simulation. We picked the North Atlantic because it is an area where we expect a lot of significant changes due to climate change, and also happens to be an area where there is a lot of validation data. So this simulation is in 1/9 degree, but future work will extend this to higher resolutions, like 1/27 degree, like 1 or 2 kilometers or so. So what we did is we added 2 new zooplankton into the cobalt model. Cobalt has these 3 size classes of zooplankton, these 2 crustacean mesozooplankton, and so we duplicated those crustacean mesozooplankton but made them migrate. And they're totally dynamic, so they're competing with the other zooplankton. The medium zooplankton are migrating shallower than the larger zooplankton. They're tracking a different isolume, but they're consuming during the day. They're residing at depth at night. And because, in our first simulations, they were kind of all they're all dying out. One of the things that we added that was really important was visual predation. So they are protected from predation at night. And this is following work by, again, Daniele Bianchi in 2013, where he first introduced this visual limitation factor for vertical migration. And when compared to ADCP observations, this model does quite well. Here we're using ADCP observations from the North Atlantic only, and the observations are in blue and the model is in red. So you can see during the spring bloom, we're tracking the observations with the model quite well, so we have confidence in this model. And then one thing that we found that was, we added this figure in here because Jamie at the beginning of the symposium showed the idealized vertical profiles from the different pumps. what our results are showing is that because the zooplankton is spending half their time up in the surface, half their time at depth, but also respiring as they go down, egesting as they go down, that the contribution for the vertical profile of the carbon fluxes from these zooplankton are actually still quite surface-oriented. They're a little bit shallower than what you might see for the gravitational pump, but they're certainly not only focused at 300 or 400 meters of depth. So, Adrian, we would like to propose a revision to the idealized vertical profile. And with this model, we can start to decompose the different effects. So we can start with the isolume depth in clear water, and then look at what the chlorophyll effect would be in reducing light attenuation, and as well as the vertical transport effect from convection. So Mathieu ran this global warming experiment in his idealized model. And here, what we can't do is we can't look at the sea ice coverage. And because this simulation is only of the North Atlantic, we're not really going to be able to look at the contribution of oxygen. But what we can do is start to pick apart the various drivers from primary production influencing zooplankton biomass, migration, fraction of migrators, and size distribution, as well as ocean temperature influencing metabolism. So bringing in together the various drivers that I introduced in the first part of the talk to look at the differences in both the depth of the fluxes as well as the total rate. So let's start with a warming experiment. So we simulated, we increased radiation and simulated an RCP 8.5 experiment. And as Mathieu showed earlier, there is a decrease in nitrate due to increased stratification. There's a higher decrease at 100 meters than there is at surface, but proportionally, the decline in nitrate is greater at the surface. And associated with that, there is about a 2.3% decrease in MPP. Because of the decrease in MPP, we then saw a decrease in the migratory biomass for the migratory zooplankton biomass at the medium size range, as well as the large size range. But we saw that the large mesozooplankton biomass declined much more. I'd just like to note that these y-axis bars are quite different. So even though you're seeing this like 5 milligrams of carbon per meter squared decline, this is a large decline here compared to the medium migratory biomass. So the community composition shifts towards the smaller size range. Because the community composition shifts towards the smaller size range, then we would expect that the community as a whole would target shallower isolumes. Simultaneously, because of the decline in NPP, we would also expect a clearer water and deeper penetration of light, so isolumes would deepen. So then the question is, how do these effects interact with each other? So this is where it gets really interesting, because it turns out that the effect of the isolume deepening is the stronger one. So even with a significant shift in the mesozooplankton community structure, we see the migration depths deepen. It's about 10 meters or so. So depending on how you think how significant you think that is, but we see a distinct deepening of the migration depth. So that's interacting effects 1. And the second one would be focusing more on the temperature effects on metabolism. So again, warming the oceans. And so what results is both particulate and dissolved. So this is DOC and DIC. Sorry, this is an error in the figure. Here, this plot shows the POC, and this plot shows the dissolved carbon, DIC primarily, but also DOC. And the carbon is released at shallower depths because of the increased metabolic rates. So this is despite the overall deepening of the migratory biomass. And then the declines in MPP and associated zooplankton biomass then also results in declines in both particulate and dissolved fluxes. This is about 5% or so total. So not huge, but still quite significant. So the combined effect of both where the carbon is injected and released, plus the reduced fluxes, would imply that there would be significant decreases in the strength of the active flux with climate change. So this is only in the North Atlantic. I think the story will change once we look at the whole ocean, but this at least gives you like one step of the way towards addressing this question. All right, so to wrap up, so what I hope that I did in this talk is to make some of these feedbacks a little bit less uncertain. I think they're still quite uncertain, so I wouldn't change this figure, Steph. But hopefully it provides a little bit of context as to what drivers are doing what. So I suggest the first order driver is MPP, but the second order effects will interact with each other in different ways, including optical penetration, plankton size structure, temperature, and oxygen. I didn't talk at all about tertiary effects, food web interactions, maybe triggering reverse vertical migration. Those are even more unknown, and I think are definitely worth studying. And then we have a first attempt at trying to model multiple size classes of vertically migrating zooplankton in an ocean biogeochemical model. And I think that for research purposes, we really do need these models, and we need them at different size classes, and we need to look at these, be able to look at these interacting effects to answer this question. So with that, thank you for your attention. Thanks for making it to the very end, and I'll take any questions.
Awesome, that was great. My God. Any questions? All right, Alexis first.
Thanks, Jessica. That was great. So I'm going to kind of play devil's advocate here, but if future conditions are showing that smaller sized organisms are going to be more preferable, do you think that a small size class should be represented in your model?
A small size class of
like the migrating organisms?
Well, so yeah, I called it medium and large, but it's really, but the naming of that is because we're borrowing from the naming of the zooplankton classes in cobalt. So the small zooplankton is a microzooplankton. The medium zooplankton is like a small mesozooplankton, and the large is would be like a large mesozooplankton, like a large copepod, predatory copepod, and krill. So yeah, I called it medium and large, but it's really just small and large. Yeah, that clarifies.
Heidi? Hey, over here. Hi, Jessica.
Oh, yeah.
Thank you. I think you said that in the simulations when you showed the smaller phytoplankton going forward, that there was an enhancement in the relative importance of the large pelagic tunicates.
Yeah.
And was that relative to all the grazing? And what happened? Why didn't the small pelagic, the appendicularian-like class, what did they do?
So I didn't show the relationship between the appendicularians and chlorophyll, but the appendicularians have a much closer, the relationship between appendicularians and chlorophyll is closer to the mesozooplankton than the salps. So they're found primarily in the coastal areas, sorry, quite a ways back. Here I cut off panel A of this plot. But in panel A of this plot, I show that the relationship between the biomass of appendicularians and surface chlorophyll is quite closer to that of crustaceans. So we think that, and these simulations are not a climate simulation, so we haven't fully run that out yet, but we think that based on this relationship, that the large tunicates will dominate over the appendicularians in a future ocean.
So that's a statistical thing from the observations, not
This
from
is a statistical
mechanism in the
relationship
model.
from the observations and one that we reproduced in our model.
I don't want to dominate if somebody else has a question, but what there isn't one, I'll keep going. What do you have a sense of what mechanisms in the model are leading to the appendicularians not responding as strongly?
So it's a combination of their metabolic rates, their food preferences. So here what we have is a dominant predation from the large tunicates on very small phytoplankton. They also, the tunicates also, these tunicates also have a really high metabolic rate because they're constantly filter feeding. And so their basal metabolic rate, I mean, there's no true basal because once you stop them from moving, they die. But their basal metabolic rate is much higher than the crustaceans, and also in the physiological data compared to appendicularians of the same size.
Jessica, thanks for your talk. Were you surprised at how sensitive this sort of cascade of effects was? Because it's only a 2.3% change in NPP.
Yeah.
It seems quite remarkable, just the sort of knock-on effects.
Yeah, I was definitely surprised. I was very surprised at who won out when we looked at the effects of shifting community size structure versus the deepening isolumes. And I think, yeah, I mean, the models also show that the MPP will change much less compared to the mesozooplankton, the kind of the zooplankton biomass, and also the fish. So the fish MIP models show declines up to 40%. The variation is quite large. So we think that this is kind of a trophic amplification effect.
Do we have more time for questions?
Yeah, sorry, I'm working on the breakout group.
Okay,
You're fine.
right. All right. Well, in this case, Debbie.
So then if you add pelagic tunicates in, so we've got small, medium, large, and pelagic tunicates, what do you think will happen? How do you think that will change?
That's a great question. Well, so I think that the mesozooplankton community will shift smaller. The pelagic tunicates will kind of counteract that a little bit. But so we could actually see more deepening of the migration depths. It kind of depends on the relative competition between the pelagic tunicates and the crustacean zooplankton, kind of in a future warming scenario, something that, yeah, I would like to run. But yeah, it could increase, and could increase substantially the active flux, if salps and pyrosomes end up dominating the ocean.
Yeah, pyrosomes.
So we now have a model with salps and pyrosomes, but no vertical migration. We have another version of the model with vertical migration. We haven't put them together, but it's not trivial, but it's not hard, yeah, to kind of now put them together. And yeah, I think it's something that I think we should work on next. Yeah.
Sounds great.
Thanks.
Any more questions? I think that's it then. Awesome. Well, thank you so much again
Thank
for the
you.
talk. It was great.